So shall we start the meeting? Welcome one and all present here. Over to Aparna.
A very happy evening to one and all present here. On behalf of the Department of Computer Science, University of Madras and MHRD RUSA organizing team, I am glad to extend my heartfelt welcome to everyone for this day 4 session of 15 days workshop on analytical tools for image processing. We all had a very useful and knowledgeable sessions for the past 3 days. In that row, we have today's session on the most desirable topic, computational modeling of cancer genomics data for targeted therapeutics. For making today's session more informative and interesting, we have with us Dr. G. Kumaresan, Professor, Department of Genetics, Madurai Kamaraj University. Welcome you, sir. I hereby welcome all the professors from University of Madras, other universities, affiliated colleges, other colleges, research scholars, and student participants for this program. Now let me call upon Principal Investigator Dr. P.L. Chitra ma'am to share a few words.
Happy evening. I am happy to meet you all in the third day of MHRD RUSA 2.0 Biomaterials 2 weeks workshop on analytical tools for image processing. And it's my inbound duty to welcome one and all present here. First and foremost, I'm happy to welcome our eminent resource person for his valuable time with us. Today, World Cancer Day. February 4th, 2022 is World Cancer Day. I light this candle in honor of all cancer survivors, fighters, and those who lost their life to this disease. Keep this candle burning to build momentum towards cure. Together, we are unstoppable. I think cancer is a hard battle to fight alone or with another person at your side, but I will say having someone to pick you up when you fall, stand by your side through every day. One day at a time, one stop at a time, do what you can do your best, and let God handle the rest. I wish our research towards cancer may help the society and help the patients to overcome with better health and love. Let us stand by the cancer patients through our research contributions and make their every day a better day. I hope and pray this forum will come to viable contribution towards the mankind. Today, we have an eminent resource person, Dr. G. Kumaresan. He is the right person of the Cancer Day. He has done a lot of contributions towards cancer, computational modeling of cancer genomics, data for targeted therapeutics. He is going to speak today and addresses, and he will throw light on this topic. Sir is a professor and head Department of Genetics from Madurai Kamaraj University. I take this opportunity to recap our last 3-day session. First day, Professor Dr. S. Santosh, MD, Nuclear Medicine, has thrown the light on cancer imaging and therapy and how using radioisotopes and how the various modalities have been used. And he has given the real use cases of how the treatment, diagnosis, everything is given. He has given us a clear picture about it. Second day, Professor D. Augustine has pointed out what is cancer, comprehensive care right from prevention, diagnosis, treatment using nuclear medicine, radiotherapy, medical imaging, surgery, pathology, and clinical laboratory. And he has also mentioned about the role of imaging in cancer care, nanomedicine applications for cancer therapy. Day 3, Professor Chandra J. from Christ University has explained the use case of respiratory motion prediction of lung tumor using artificial intelligence. He has explained about how the images have been captured and how the area of the affected portion has been measured with the help of the logarithmic transformations, power law transformations, and she has given how artifacts can be handled out, and she has given the complete picture about the prediction of lung cancer using AI perspectives. So today we have our eminent resource person of the day, Dr. G. Kumaresan, Professor, Department of Genetics, Madurai Kamaraj University, to enlighten us on computational modeling of cancer genomics data for targeted therapeutics. Sir has contributed towards cancer patients with a lot of work. Today is a World Cancer Day. Again, I'm just insisting. We are honored to have with you, sir. Welcome, sir, for this program. Sir is the right person, resource person today. Over to Aparna.
It's a pleasure for me to introduce our eminent resource person, Professor Dr. G. Kumaresan, sir, head of the Department of Genetics, Madurai Kamaraj University. He is a gold medalist in UG and PG and done his research doctoral degree in Madurai Kamaraj University. He has received many awards and honors, including Dr. M. Varadarajan Memorial Endowment Prize in 1997, BTM Ashi Murray Grant Award in 2004. He has authored more than 40 research papers, 5 research manuals, 2 book chapters, and a patent. All his research papers are published in highly reputed journals. To name a few, delineation of gastric tumors with activated ERK-MAPK signaling cascades for the development of targeted therapeutics in the journal Experimental Cell Research. YY1 regulated transcription-based stratification of gastric tumors and identification of potential therapeutic candidates in the Journal of Cell Communication and Signaling. He has guided 8 PhD scholars and 6 more scholars are currently pursuing under his guideship. He has trained 75 research fellows. Sir have completed 10 research grant projects and involved in 5 more ongoing projects. All his research works are with societal cause and highly beneficial. For example, his ongoing project, Comprehensive Immunological Profiling to understand the prophylactic potential of selected Siddha medicines to combat the COVID-19 pandemics, is dealing with the recent situation. A day is not enough to speak about his research works and achievements, as all are eager and excited to learn and interact with him. Without any more delay, I humbly hand over the session to the professor. Participants, kindly mute your audio and you can post all your queries at the end of the session, or you can also ask in the question answer session at 5:15. Over to you, sir.
Yeah, thank you very much, ma'am. And I would like to thank Professor Chitra for giving this opportunity. And incidentally, this also being a cancer day, and then that's a good coincidence. And yeah, as mentioned by Professor Chitra, yes, we all those who are working in cancer should make extra efforts to make sure that something is contributed. And yeah, so hope my slide is visible.
Yes, sir. Visible, yeah. Sir.
Yeah. So good afternoon, participants. I understood that many of you are from various divisions of computer science, but mine is basically a cancer biology, and then but still I'll try to touch upon what kind of computational methods we are using. But I am definitely not an expert in computational aspects, but in the biologist, but we are using computational modeling. So I'll explain you what kind of work we are doing on computational modeling of cancer genomics data. So basically, it's though it is cancer, the primary starting point is human genomics, and you all know very well we have 23 pairs of chromosomes, and then 15 years back people started sequencing the human genome. They sequenced the human genome of a single individual, and it was costing maybe a few $1 billion. At that time, to sequence a single genome, it was a massive effort, and then people took 6-7 years and then sequenced a single genome. Now we have, now if I want to sequence my own genome, I can sequence in some 30 to 50,000 rupees. So the technology has become very cheap. And now, you can go to ncmdl.org or the other browsers where you can get the sequence of thousands of individuals. So 10 years back, we were in a stage that the human genome was sequenced. And then now, people started sequencing everything. For example, in many countries, people have done 1,000 genome projects or 1 million genome projects, which means people are collecting the genomic DNA of 1 million individuals in the country, and then sequencing everything, whole genome. Our genome is for because I believe still a part of non-biology participants should be there. ATGC, the I'm sorry, just hold on. I'll be back in a minute because I have an important call from RUSA office. So I'm really sorry. I'm also being the RUSA coordinator of Madurai Kamaraj University, and then we have an inspection Monday, and then I am getting repeated calls from RUSA. Sorry, really sorry for that. And yeah, so ATGC is one base is English. We have 26 letters and we have a code, and similarly, our DNA code, our genetic code is made of ATGC. We have A is a single base. We have 3.4 10 power 9 bases in our genome. So that codes for the entire biology, and that is responsible for the diseases and including the cancer and everything. So understanding the genome sequence is very important. That's what I told you. Sometime back, 10 years back, people sequenced with $1 million expenditure, but today it is possible to sequence with 30 to 50,000 rupees. And even in RUSA grant of Madurai Kamaraj University, we are doing a 100 genome project from Madurai, some selected diseases. So what I'm trying to say, what I'm saying is sequencing is very cheap now. But the problem is analysis is very costly. You can imagine 3.4 10 power 9 base pairs. One data would be sequence from one individual would be maybe somewhere around the 1 terabytes. So now computer science students can understand that if now in our own projects, we are analyzing the genome sequence of the humans from all over the country, because we are doing a project, we are trying to understand the migration, evolution, and then some disease specific genes. Now, for example, if we say diabetes, people have sequenced some 50,000 individuals whole genome sequence from diabetes individuals. Now that can be analyzed. See, now the point is all the data, whatever the human genome sequencing or disease genome sequencing, all the data is there in the open source. And any researcher can take the data, analyze, reanalyze, and then try to make their own model, then make their own findings, or they can test their own hypothesis on the data available in the public domain. So that's what now, one in a biologist can't be successful unless and otherwise he has some computational skills, because it's no more a laboratory thing. It's generating a data. For example, if I'm going to isolate DNA from an individual and then sequence, that is a technician job. But the real scientist job being analyzing the data. But to handle such terabytes of data, you need to have computational skills, computational tools, computational knowledge, and again, you should be able to do some computational modeling. So we are basically, I'm a biologist, but I'm trying to learn certain things, and then many times we learn through students and then do everything. So now all the genome sequencing projects, 1,000 genome projects, everything is available in the open access. Yeah, in human genome we have 20,000 genes, but I don't know how many of you are from non-biology background, so I can change my tune actually a little bit. Are there, madam? I don't know how many of them out of 100 participants, I don't know how many of them would be non-biologists. Any idea, ma'am?
Almost 75% of them are non-biologists, sir. But anyway, we would like to have this type of things in our forum, sir. Since we need to understand all this genomics, I think you can proceed, sir, and you can give some basic level introductions and then proceed, sir.
Yeah. So, for example, now, as I told, we have 3.4 10 power 9 base pairs. People used to say 3.4 GB. GB is our genome size. But that is a huge. But we have genes. Genes means, for example, insulin is a protein. It is involved in governing the diabetes. That is a single gene. Likewise, in human genome, we have 20,000 genes only. So genome is so big, but only 2% of our genome is coding for biological information. So we have 20,000 genes. Like insulin is one gene. Similarly, we have 20,000 genes. For example, thyroid hormone is one gene. Likewise, we have 20,000 genes coding for 20,000 proteins in the human genome. Now, one way is like analyzing the 3.4 GB data, 3.4 GB of bases in every context. That will give a kind of information. And another one is you can analyze 20,000 genes because the in English we have 26 letters, but you can have form so many words. And similarly, but here it is not like that. We have a lot of bases, but we have very few number of genes. 20,000 genes is very less, which is contributing for 20,000 proteins. Now, there are some complexities. The central dogma of molecular biology is from DNA, you will get protein, and the proteins will be involved in function. In a cell, you'll have nucleus, you have different organelle, you have so protein can present in different places and all those things. Let us forget about all those things. Just remember that we have 3.4 10 power 9 bases and we have 20,000 genes. Let us forget about all those things. Now, 20,000 genes is a decent number. And this gene expression, we can always model in different contexts, because classically, one PhD student will be analyzing one gene. For example, insulin gene, will be analyzing for 5 years and then try to make that as a PhD work. So you can classically in biology, people will be analyzing one gene at a time. But now in genomics, genomics is a field where you can analyze all the genes at a time, because now computationally everything is possible. And now there are some methods available to analyze the expression of all the genes. For example, from a cancer tissue, you can isolate RNA and then analyze the expression of, expression means from DNA, you have whether RNA is there or not, that is called expression. You can analyze the expression of all 20,000 genes from a few micrograms of RNA. And then you can do a lot of modeling. One of the modelings is you can see the first top left figure. The pink, each line represents a gene's expression. You can hear each column is a different sample or a tumor sample. You can see all the pink lines are having uniform expression. Classically, while we are biologists, we never had a chance to look the data in this way. But now, being while you can see the data in computationally, or where you can model computationally, then you can get a very good understanding. From this, you can understand that all the pink genes, they are related genes, they are involved in the same function. For example, that's what I told you. If I want to understand the function of a gene, it may take 5 years for me if I want to do experiment biologically. Maybe I have to develop a mouse model, or I have to grow the cell lines in the laboratory, and then I have to do that. But now, once we have this kind of genomics data, which is there in the open access, and you have the expression information of 20,000 genes, then you can try to get the model all the pink genes are involved in the function. So that is this is a cluster, and all these pink genes are involved in the same regulated by the same factors. And this is one of the classical way by which you can model a data. And another one is we have 20,000 genes, but now all the 20,000 genes, but we don't have 20,000 processes in the human system. We may have 1,000 processes in the human system, or few hundred cellular processes in the human system. Now 20,000 genes can be grouped into processes or grouped into biological functions based on some computational modeling. So from that we can try to understand, because otherwise The problem is you will have the expression of information of the gene expression, but you will not have the functional knowledge, what that gene is contributing, probably what is its relationship with the other gene, probably what is the tissue in which that gene is expressed. But now by this kind of computational modeling, we can try to understand that the genes involved in a particular function, genes involved in a particular biological process, and what is the very important gene. People call it as a node and hub and so on. So in our laboratory, what we are trying to do is the top figure is here. Each column represents a cancer sample, and each row represents a gene. As of now in clinic, in any cancer, we have only some 2 or 3 subtypes are known. For example, in gastric cancer, only 2 histological, because the doctors diagnose the cancer based on the tissue histology, like how the cells are arranged, what is the tissue architecture. Based on that, doctors used to decide what is the cancer subtype. Because cancer is not a single disease. For example, if we take any cancer, breast cancer or like stomach cancer, it's not a single entity. It is a heterogeneous thing. There may be so many types possible. As of now, the reason for the failure of the disease, therapy is a particular drug will not work for 100% of the patients. It may work for 50% of the patients, and it will not work for another 50% of the patients. The reason is that 50% belongs to a subtype, another 50% belongs to some other subtypes. But we are giving the same drug for everybody. So now we have the information of the gene expression here, each row represents a gene. You can see that this is stomach cancer. It can be stratified into some ABC, some 8 subtypes. For example, this A subtype, you can see some genes are expressed in this subtype, whereas in subtype B of gastric cancer, you have some other genes expressed. Okay, so in this subtype, some genes are expressed. In this subtype, some genes are expressed. In this subtype, some other genes are expressed. So biologically, based on gene expression, all the stomach cancers are not same. So in different, you can categorize. As of now in clinic, only 2 subtypes are available. But now computationally, based on genomics data, you can try to classify cancer into maybe 8 subtypes or 10 subtypes. So for that, genomics information is very much useful. Now that's what I'm telling you. If you give from a single sample, you will get terabytes of data. That data you can model and you can analyze and you can try to classify cancer into multiple subtypes. Okay. Now here, as you see here, based on this gene expression information, gastric cancer can be classified into 8 subtypes. Now, what is next? First thing is in clinically you have to need to have a method to diagnose as which subtypes this one is, and subsequently you need to understand what drug would work for different. Because as I told earlier, a single drug will not work for all the 8 subtypes, but one drug will work for subtype 1. Another drug may work for subtype 2. You may have to identify another drug for subtype 3. Likewise, we have to, based on genomics knowledge, we can subdivide cancer into multiple subtypes. That is one point. That is point number 1. And second point is, for different subtypes, we can develop different drugs or different combinations of drugs. That is point number 2. So if we do that in future, as of now in clinic, only 2-3 subtypes are known. In future, we'll have 10 subtypes in a cancer is possible. Only once you have 10 subtypes, then you have 20-30 drugs in different combinations. In different combinations can be fixed for different subtypes. This is called targeted therapeutics in cancer or personalized therapeutics in cancer. And all these things are possible only by computational modeling. And this shows like that, you have each column represents a gastric tumor sample. Gastric tumor is nothing but a stomach tumor, and here some 200 columns are there probably, and each column represents a patient, and each row represents a process. Sometime back I was talking about genes and then some biological processes. Similarly, we have something called the pathways. Pathways means, for example, some 100 genes join together and then they do a function that is called genes involved in the pathway. So because Genes, will you in clinic, will you target a particular subtype of a cancer? Targeting a gene may be difficult, but targeting a process, targeting a pathway is possible. So these are the pathways like, MYC, stem cell pathway. NF-kappa B pathway is involved in inflammation. You might have heard about the inflammation is a common problem in many diseases. Now, the pathway involved in inflammation is NF-kappa B. Okay, so now for the point is, now you can see in this particular set of cancer patients, these pathways are highly expressed. In this subset of cancer patients, these pathways are expressed. Now, if you have different drugs, so far we are talking only about DNA, genomics, genes, pathways. Now we are seeing how a drug can be connected to this model. Because here, like, here whatever you have shown here, it is, for example, saliracid, and these are drugs which are in clinical use or in clinical trial. And for example, this drug can be used to target these pathways, which means which can use to target this subset of cancer pathway, cancer patients. And similarly, this drug can be used to target these pathways and this subset of cancer patients. So this is the model of personalized medicine or genomic medicine or targeted therapy. And throughout the world, many researchers are working towards that, and we are also working on that aspect. Sorry. So in our laboratory, we what we have done is by using cancer cell lines models. These are the cell lines, and you can see these are the cancer cell lines from the cancer patients. These cell lines are basically derived, and this can be cultured in the laboratory. In the laboratory, we are growing the cancer cell lines and using that as a model, because sometimes you need to have a model system for any experiment. And what we do is like. We grow the cell lines and then you activate, for example, these are different pathways. NF-kappa is a pathway, NFAT is a pathway, HIF1 alpha is hypoxia. In COVID-19 also, you might have heard much about the hypoxia signaling. And ESR is estrogen receptor. This is one of the female hormones, and it is working in most of the in breast cancer and also in some other cancers and also in gastric cancer. And the PPR gamma is a kind of a PPR is a pathway which is involved in many diabetic things. And hepatocyte nuclear factor HNF is involved in liver, normally in liver tissues. Now these pathways in cell lines we have activated or we have inhibited and then identified the genes, and so we have a mechanism by which we can understand the pathways. So what we have done so far in the laboratory is, again, this is a heat map which shows the activation pattern of cancer pathways. Again, all these involves computational methods, and then gene expression is basically, so what we do is you take a piece of cancer tissue from the patients, because doctors dissect out the tumor during the surgery, and that can be collected in the medium, and then we bring it to the lab and then analyze we'll isolate RNA from the tumor, maybe from 10 mg of the tumor, you can isolate RNA, and then particularly subsequently the RNA can be subjected for whole RNA sequencing, NGS methods, next generation sequencing methods, or there are some microarray profiling methods are there. Using them, we are profiling the understanding the expression of genes in a particular tumor. Likewise, here each column represents one tumor sample. Okay, some probably saying some 300 tumor patients. So some of them are data available in the open access. Here 611 gastric tumors, and here in this another cohort 438 gastric tumors. And these are the data available in the public domain. We took the data and then we have done a pathway modeling and then try to understand that what pathways are activated in which subtypes of diseases. And then we have, for the first time, gastric cancer, we have made an attempt to classify gastric cancer into 8 subtypes. That's what denoted here. One, you can see in cancer subtype 1, subtype 1, you have these pathways activated. For example, lipid process is highly activated in subtype 1, and MTF is highly activated in another subtype 2. And likewise, in subtype 1, some pathways are highly activated, and in subtype 4, some other pathways are activated. For example, in subtype 8, multiple pathways are activated. So you can subdivide cancer into multiple subtypes. That's what we have demonstrated. Now, in all these subtypes, we know what all are the pathways activated. Once you know what all are the pathways activated, then pathway specific chemicals or drugs are available. So then we can say that for subtype 1, you can use these pathways. So that is the way in which we are working. And there's like for example, subtype 1, we have defined as these pathways are highly activated: MTF, Notch, NFAT, and so on. In subtype 2, these pathways are activated, and also some other pathways are also activated. Some of you might have heard about the interferon pathway, proteasome pathway, PPR gamma pathway, estrogen receptor pathway. So different combinations of pathways are activated in different subsets of the disease. I will not go into that. Okay, I think should have shown this slide. I think normally, in stomach, we have cancer. We'll dissect out. As of now, in clinic, the clinicians are classifying the cancer based on the histological staining. And then, but now what we have done is we have isolated RNA from them, and then we have done the whole genome expression profiling. And then, we are trying to group the tumor as tumor, normal, and which subtype, and so on. Okay. So by now another aspect now I have I'm sure that I have made you to understand what is genome sequence, what is gene expression, what is a pathway, how a genomic medicine or targeted therapy or personalized medicine is possible in cancer, and all those things in one side. On the other side, in biology, now, as I said earlier, we have a lot of data. Now we have publication policy. Whenever you are going to publish a biological data, the data has to be first placed in open source or at least in a database. And then only in future, anybody can take the data, because in long time back, whatever I do is science, but now it is not like that. I have to put the data in the deposit in a repository, and then I have to give the access to the public, and then only many good journals will accept any work. So that's what now the open sources, resources, databases are filled with lot of information of the cancer patients, cancer tissues, and so on. And now that is what in one way the research has become easy. Mainly if I have some computational power, then I can take the data and then analyze them. So what we have done from Madurai is we have collected some 50 samples from Madurai Meenakshi Mission Hospital, and then we have done a whole genome expression profiling, and then again we subdivided them into various subtypes, subtype 1, and we found that subtype 1 is very common, subtype 8 is also very common, and the subtypes are less common. So 1 and 8 are common, other subtypes are moderate. So this kind of informations are varying in different countries, in different cohorts of the disease, and this is information from Madurai. Okay, so this is what we have done in the laboratory with reference to the gastric cancer data. Now I'll change to another modeling, what we have done in a hepatocellular carcinoma a few years back. And normally a gene expression will be like this. You can see each row represents a gene expression. Red means higher expression of a gene. Green means lower expression of a gene. Because you have compressed the information of 200 genes expression across. I'm sorry, I'll just be back in a few seconds. So now here each row represents a gene and each column represents a tumor sample. Now you can see LD1 is a set of genes. They have higher expression in some set of patients. Whereas you can see LD2, this particular set of genes, they are expressed in some other tumors. Now, it is possible to group the genes, or it is possible to group them as pathways which are expressed. Now, for example, from this, what we have modeled is based on the expression pattern of the genes or pathways in subset of tumors. Computationally, while you analyze their expression pattern, the pathways which are relatedly expressed together, they are clustered as a single set of pathways, the LD1 pathways, LD likewise different pathways. Now, from this, what we understood is, for example, this particular set of pathways, this is just based on the this is called co-expression modeling. So whenever all the genes are having same expression pattern, for example, people used to say, now you tell me your friend, I will tell you a character, likewise. So what are the genes expressed in the same way, if you could understand and then try to predict their function, because some of the genes are well known. So but here, instead of genes, we made this as a pathway. Now you can see here, LD1 pathways are highly expressed in the estrogen receptor signaling is highly activated. And similarly, the drug rosiglitazone is really activated in this subset. So likewise, we can try to categorize the pathways. We can try to link the drugs with the pathways. For example, estrogen receptor is a protein which is highly expressed in ovary. It is also expressed in breast cancer. It's a female hormone. Now, in breast cancer, estrogen receptor is playing a big role. In 30% of the breast cancer patients, estrogen receptor signaling is playing a big role. And then what people have identified is they developed a drug or a chemical which can block estrogen receptor. The drug is called tamoxifen. Now, in the breast cancer patients where they have higher expression of estrogen receptor, if you give the drug tamoxifen, then the tamoxifen drug will bind with the estrogen receptor protein, and then that will block the cancer growth. Okay. Now in this module, you have the higher expression of estrogen receptor. From this, the inference is, by doing a computational modeling, you are trying to say that estrogen receptor is playing a role. Now, you know very well, based on the background, whatever I have told, tamoxifen is a drug which can be used to treat the breast cancer or any cancer which has higher expression of estrogen receptor. And similarly, rosiglitazone is a drug which is a diabetic drug. And now that drug also can be reused, repurposed for this subgroup of cancer patients. So now, what I'm trying to say is, we do a simple computational modeling and then try to understand the pathways. We try to understand the drugs which are related to that. By that way, we can connect the drugs, cancer subtypes, we can connect the pathways, and so we can talk about not only the diagnosis, we can also talk about the therapy, we can talk about the targeted therapy. This is the way by which a co-expression based modeling can be used for cancer therapy, mainly for targeted cancer therapy. And similarly, in this module, if we see, you have interferon signaling is there, you have hypoxia signaling is there. Now, after COVID-19, everybody knows about hypoxia. If you have a drug available to treat hypoxia, the same drug also can be used for this subset of hepatocellular carcinoma patients. And similarly, in another subtype, you have some other pathways like proliferative pathways. You can use those drugs to basically treat particular subset of drugs. Okay. So by this way, you can connect drugs, based on computational modeling, you can connect to the drugs. I think that's what, as I mentioned earlier, you have a proliferative cluster, proliferative pathway, and then you have a hepatocyte related pathway, and you can do all these things. So we have made a model based on this data. Normally, in a normal liver, you have estrogen receptor pathway is activated, hepatocyte nuclear pathway is activated, and everything is there. And then subsequently, will it go in the early stage of liver cancer in cirrhosis, many inflammation pathways, inflammatory gamma, and NF-kappa B, and many inflammation and hypoxia, HIF1 alpha is hypoxia signaling, they are getting activated. And subsequently, when it becomes advanced cancer, the pathways like AKT, WNT, MYC are activated. So now, based on the disease stage, you can use different inhibitors, different pathway specific inhibitors to target them. Okay, so this is part of the work what we have done in hepatocellular carcinoma. And then, as I said earlier, one of our areas is we are trying to work on the Siddha medicines, homeopathy medicines, because for allopathy medicines, we do know the mechanism, molecular mechanisms. We do know exactly the target. But for homeopathy medicines, we do not know the target. Siddha medicines, many times we do not know exactly what is the biological or molecular model, molecular target. So what we have done is we took some homeopathy medicines, because some time back I mentioned you NF-kappa pathway, the inflammatory pathway is highly activated in hepatocellular carcinoma, a subset of hepatocellular carcinoma. And then we took a cell line model, and maybe I will not go into that. We have developed a cell line which can be cultured in the laboratory, and then you can grow in 96 cell plate. In each well, you can add one drug and then try to see whether the drug is inhibiting NF-kappa pathway. So by doing so, here each dot represents one homeopathy drug, and we could identify a drug called aripiprazole. It's a homeopathy drug which extremely inhibits the NF-kappa signaling pathway. So from that, one can infer that, as you know, NF-kappa pathway is activated in cirrhosis or a particular subset of hepatocellular carcinoma, and then you can use the drug aripiprazole to that particular subtype. So by this way, we are doing genomics, computational modeling, and also experiments in the laboratory, cell line models, and then trying to connect the traditional drugs like homeopathy medicine, Siddha medicines to particular subset of cancer. And this is similar co-expression modeling what we have done in breast cancer. In breast cancer, there are some 3 subtypes. One is triple negative, as I mentioned, estrogen receptor positive. In those breast cancer patients, estrogen receptor signaling is playing a role. As I said, tamoxifen can be given for the patients. In another subset, you will have progesterone receptor will be playing a big role, and then you can give a drug. In another subset, there is a protein called herceptin, and that will be highly activated, and you can give antibody for that. But there is another subset in breast cancer. People call it triple negative. It is negative for estrogen receptor, negative for progesterone receptor, it will be negative for herceptin receptor. In those cancer patients, normally the problem will be they will be very bad, and then disease progression will be fast, metastasis will be more, and response to the drug will be less. And now we took those patients' mRNA gene expression data and then data co-expression network modeling, and we tried to classify those patients into 5 subtypes. See, triple negative breast cancer itself is a subtype, and now we have subdivided triple negative breast cancer into 5 subtypes. By doing so, now you can go, as I said, triple negative breast cancer has poor prognosis, and now that can be subdivided into multiple subtypes. And for example, you can see MET pathway, MYC pathway is involved in a subset, interferon pathway is involved in a subset. Now we can go for targeted therapy for these particular subset of patients. This is what a breast cancer patient, triple negative breast cancer patients, we could show they can be classified into 1, 2, 3, 4, 5 subtypes. So now we know what are the pathways activated, and we can go for a targeted therapy. We have made a biological model. For example, in one subset of breast cancer, triple negative breast cancer patients, proteasome pathway is highly activated. Proteasome is a pathway, oncogenic pathway or a cancer pathway, but people have already identified drugs for that. I think chemists have already identified drugs for proteasome. Now there is a drug called bortezomib. If you get the bortezomib drug and then just add into the cells or give to the patients, that would work well. So likewise, we are trying to subdivide a cancer into multiple subtypes and then understand the pathway and then link with some drugs, and so we can go for a targeted therapy. We're also doing similar modelings and trying to understand the metabolic pathways, and then maybe I'll stick that. So histochemically, if you see the histopathology of the tumor could look like this, and the clinicians will make a decision based on this kind of the pathological information. But in the current work, what we propose, in all over the world, people those who do genomics, why don't we use genomics data, gene expression data as a marker for diagnosis? I think these slides are getting repeated, and this is what the cell lines would look like. And we can do a lot of assays in the laboratories. And being the non-biology students, I don't want to talk much about that. So we have developed a lot of cell lines in the laboratories, and we screen thousands of drugs. For example, in one of the recent works, We screened 1,280 drugs against a hypoxia pathway. And from that, now we know that what all are the drugs which are inhibiting the hypoxia signaling. Now, sometime back, I have shown you how hypoxia signaling is playing a role in a particular subset of a disease. Now, you can take these drugs which are inhibiting hypoxia signaling and to say that for that particular subset of disease, we can use this. Okay. Of course, we are also trying to identify, these are different methods by which you do research in the laboratory. You can try to understand the expression of a protein in the cell. This is, the green represents the expression of, for example, after adding a drug, you are seeing the loss of expression of the protein. And we are also by doing so, understanding the molecular mechanisms of the drugs. We are using a lot of drugs in clinic for various clinical conditions. But for many drugs, we know the mechanism, but still for some drugs, we do not know the mechanisms. By doing this kind of chemogenomics, we can try to understand the molecular targets of the drugs. Likewise, in another work, we have decoded what all are the pathways which are inhibited by the drug called the nimesulide and silicoxy. And we have done that, and the screening, all these things. Like once you say that, one drug is inhibiting these pathways, and then you say that across cancer patients, you can, this is what we are doing in all the work. We are trying to classify, we are trying to stratify, we are trying to group a subset of cancer patients, and for which, in which a particular pathway is activated, and then which drug can be used. This is the way by which you can connect drugs with gene expression. Yeah, and in another work, what we are doing is whatever the condiments which are there in a kitchen, turmeric, cumin, everything, you take it and then try to identify what pathways they are inhibiting, and eventually we can identify for example, M9, what is that? It's really inhibiting the cancer cells growth. M2 is myristica, it's javikai, and number 3 is nilavembu. Nilavembu is really good. I think we have recent data against the many cytokines in COVID-19 infection also. Nilavembu is really good. And we likewise, earlier for many traditional medicines, Siddha medicines, we don't know the drugs, we don't know the mechanisms. But now by doing this kind of work, we are trying to identify the molecular mechanisms. Okay, so this is all about cancer, and then maybe next few slides I'll talk about the work what we are doing recently. And you all know that in Tamil Nadu, in many places, archaeology department of government of Tamil Nadu is involved in excavations, and they are identifying a lot of urns, and in which there are some skeletons available. And we are, Maria Kamaraj University is collaborating with the archaeology department of government of Tamil Nadu through a RUSA project, and in which we are trying to isolate the DNA, because these skeletons are believed to be 2000 years old. Now we are isolating DNA from them, and then doing the whole genome sequencing, and then trying to understand their migration pattern. Because human beings, all of us are basically from Africa, and then we took different routes and then reached different parts of the world. But now, as I said, people have done 1,000 genome projects all over the world. Now, if you do the whole genome sequencing of these ancient skeletons, we can try to see that what is our relationship with the different populations, and what is probable our migration route, and all those things we can understand. And we are setting up the laboratory, and then we also can analyze the chemicals. What we are doing is we are collecting the organic materials from these urns, and then trying to find out what kind of chemicals are there. And then we are setting up the laboratory, and then trying to get the DNA and everything. So right now, we are working on human genome, and similarly, in many habitation places, people also found many animal bones, and we are analyzing the whole genome of those animals, and then trying to find out what 2000 years back, what were the animal species, and similarly the plants. Because for example, if you take rice, there are at least some 3000 rice for which people have done the whole genome sequencing. And rice, probably there are some Japanese varieties, Chinese varieties, and then so many rices are available. And now we can understand that maybe in the places like Sogalai, what was a rice variety which was used 2600 years back. We can try to find out, and we are working on that. And as I said, they are also collecting the organic materials and then trying to see. And then the bacteria also. because 2000 years back, what were kind of the bacterial load of the oral microbiome and everything we are trying to analyze. So this is the ancient genomics work which we are working right now. And maybe I'll stop here and then we'll discuss more. And I would like to thank my PhD students. And I would like to thank the grant support. I think all the cancer work supported by Department of Biotechnology, Government of India, Department of Atomic Energy, Government of India. And this ancient genomics, what I'm talking about, is supported by RUSA program of Madurai Kamaraj University, with MHRD and Tamil Nadu government. And for this hospital samples, we have collaborations with Meenakshi Mission Hospital Gastroenterology Department. So I would like to thank the all the collaborators, funding agencies, students. There is a teamwork. So I'll stop here and then we'll discuss. I'm glad to answer any questions.
Participants, you can unmute and ask your questions. You can also post your questions in the chat box.
Sir?
Yes, ma'am.
Sir, where are you collecting live data, sir? Live cancer data?
Live cancer.
Yes, sir. Cancer data, sir.
No, cancer.
Sir, I am referring. Okay. Sir, I am referring PCIA data format, sir. Data set, sir.
Okay. See, as I said, almost for several thousand cancer in different cancer types, data is available in the open source. For example, there is a portal called the TCGA or CBio portal.
Okay.
Where you have the information, and then there are so many data. Just Google it, you will find it out. And then we have made our own cohort from Madurai. That is, we have collected the samples from Madurai Meenakshi Mission Hospital. And otherwise, all are from open resources, and some of them are from collaborators.
Okay.
Sir, we have a question. Yeah. We have a question in the chat box. Purnima ma'am has asked whether there is any research focusing on Siddha medicine, breast lumps, and tumor.
Actually, in this case, we are trying to find out the molecular mechanism of the Siddha drugs. But we are trying to link the Siddha drugs with a pathway. We are trying to link the Siddha drugs with the gene expression in stomach cancer. But of course, it can be done, but I don't know exactly what people have done. But to my knowledge, Siddha medicines, that is famous only in our place. But the molecular mechanisms, and that's what we are opening up a new area called Siddha Genomics, and we are interested, and we are also proposing a grant to the government of Tamil Nadu, trying to understand the genomics and everything. But as of now, for many Siddha drugs, and even for homeopathy drugs, the molecular mechanisms are not known.
Sir, I have a question. So you were talking about computational modeling, in which what are the tools you will be using, sir, and how do you get all this genomics data with different colors and the features also you have mentioned? We people are working on artificial intelligence. We are in need of more features to make our model accurate, and we would like to have some automations. How do we incorporate these features, and how do we understand these features, and how are we going to implement in our work, sir?
No, it is a very hard question because if I know the answer, I also may try to do that because we are also interested in AI, ML, and using applications. But if I understand the question rightly, first thing is all this data is available in the open source. That's not a problem. Second thing is now the gene expression is the information of expression. For example, in one cancer patient, you will have 20,000 data points.
Okay.
So in an Excel file, you will have a single column is a tumor sample, and you will have 20,000 rows, which is a quantitative information of the 20,000 genes expression. Like this, if you have 1,000 samples, 1,000 column versus 20,000 rows. So this is the expression information. We'll have the quantitative information, and now we are trying to make them into pathways. We are trying to make them into process. We are trying to connect them with the drugs. So this is basically a genomic modeling. And there are a lot of tools in R. There are a lot of programs available to do all those things, to do all those modeling. But whatever we do is a very basic modeling, but definitely there are a lot of methods would be available. And if there is a possibility, and then we can collaborate in some aspects sometime and then do that.
Thank you very much, sir. We will come back to you for this, sir. Surely, we are trying to go with some machine learning models with respect to this. With these features, whatever you extract as a feature, I think that would be helpful to us to make a model. We'll come back to you, sir, when in a future, sir. Thank you so much for it.
There is a question how to justify and verify the computational model. Yeah, that's a good question. So whenever you do a computational modeling, and then, for example, imagine I am saying that we are identifying by computational modeling in a subset of a disease, you have a particular pathway activated. And then you can find out a cell line model in which that particular pathway is activated. And in that particular cell line, you add the drug and then see that whether drug is affecting that particular protein or that particular pathway. So by using cell line models, we can test them. By using mouse models, we can test them. And if those are successful, then people can go for clinical trial and so on. Cognitive impairment fields also. Yes, ma'am. Actually, all these things are applicable to all the fields. But the advantage with cancer is you have tons of data available in the open source. But other diseases, the data may be less, but still the data is available for almost all diseases. We are doing some other work in genome assessment studies and everything. And so it is available. I'm sure that the data is available for almost all the diseases.
Uday Kumar sir, you have raised your hand. You can unmute and ask the question.
So mathematical model, as I said in the beginning, I am a biologist. My students do all the things. So one of the models is like, network modeling, co-expression network modeling. And then we make a compress the gene expression information into compressing 100 rows into a single score, those kind of scoring into a pathway, those kind of basic biological model we do. I don't know exactly what is the process, computational process or mathematical process in that. Yeah.
Participants, you can ask your questions or post that in the chat.
Motifs available for all diseases. Motifs means, in biology, motifs, people talk about a particular motif in protein also. But, in this work, we are talking about thousands of genes expression, 20,000 genes and so on. So we are not seeing any, I don't know what you mean exactly motif, but here we are not we are trying to recognize the patterns, different patterns and so on. And yeah, not motifs exactly.
So, yeah, by motive, I mean the pattern only, sir. Is the patterns available for diseases, sir?
All these things are patterns now, because whatever the heat map I saw is the pattern. So we are trying to see the patterns unique for a subset. If you mean the motif means a pattern, yes, because the subclassification of a tumor into multiple subtypes is based on the pattern. So we are trying to see different patterns in different individuals or in different subgroups. Yes, motifs are available. That's what within breast cancer, we can try to see different motifs for different subsets. So are there then we can individualize this to individual? Those all are the areas of research. So motifs, what do you, if you mean it is the pattern, then pattern recognition is based on the gene expression. We are trying to see the pattern. Yes, the patterns are unique for a disease. But again, within a disease, we can, because data is too noisy. So we can't compare gastric cancer with the breast cancer. Within breast cancer, we can try to see different motifs and then try to assign a motif with a particular subtype, or assign a motif with a particular function, assign a motif with a particular threat. Within a disease, it is possible.
So is there any validation like the pattern which we identified is exactly the pattern which causes the disease and so on?
Yeah, exactly. Because now we do all these things in the tumor sample open source, and then subsequently, because for example in breast cancer, 50 cell lines are available. 50 human cell lines are available, which can be cultured in the laboratory. Now, if you are seeing a pattern in a particular subset of breast cancer patients, then you can try to identify the similar models in the cell lines and then try to check them. Of course, we can check them in the cell line models subsequently, and we can also develop mouse models and test them.
So the pattern verification is just with our own model creation. Or is there any data set available that these are the patterns for this type of disease?
Everybody can do their own research. They can say their pattern is there. But when you do research, we have to try to look for our own pattern. That is the best way. But of course, some already established patterns are available. We can try to see whether that is reproducible, and we can see that. Because everybody is trying to, because in research, everybody is trying to model in their own way. They just don't want to what others have done that. But anything can be done. Patterns already available also can be tried to recap.
Thank you, sir.
Thank you.
Participants, kindly fill the feedback form. Any other questions you have, you can post that in the chat or you can unmute and ask.
So,
Any questions from the audience?
So thank you very much. Thank you very much, everybody. And thanks for your time. And if you have any questions, you always email me also, and I'll be more than happy to answer. Thank you very much.
Wait, let us go for the vote of thanks, sir, please.
A sincere thanks to Professor G. Kumaresan. Thank you, sir, for sparing your valuable time and informative and fruitful. I'm extremely grateful to Professor Dr. P.L. Chitra ma'am for organizing such a wonderful program, which gives us a platform for the students, scholars, and researchers to exchange ideas, clarify doubts, and get inspiration from the field experts. Thank you, ma'am. Participants, please fill in the feedback form posted in the chat box. Let us all meet again tomorrow for another beneficial session on breast cancer image classification using deep learning. It will be a full hands-on session by Dr. M. Nachammai. Participants, please get ready for tomorrow's hands-on session with GPU-based laptop desktop installed with Python 3.6. Thank you all.
So
Gentle reminder.
I take this opportunity to thank our Professor Kumaresan sir for having accepted our invitation in spite of his busy schedule on Monday. Thank you very much, sir, for your valuable input. Most of our research scholars and we people are in need of data and features. How are we going to do some AI model based on the already existing data? So we would like to learn about this type of genomics and structures, and you have been given us a light through this ideas. Thank you so much, and we have understood, and we will have to work more on this, and we should still go long ways to go to this mile, sir. And let us incorporate this type of features and modeling extracts to our work, and let us make it a fruitful model. Thank you so much for your valuable time and valuable input to us, sir.
Thank you very much, ma'am. Thank you very much for the opportunity. Thank you.
Thank you, sir. So I would like to thank all the participants for your consistent support, and we will meet tomorrow. Tomorrow we will have a hands-on session based on breast cancer by Nachammai from Siemens. So all of you get ready with your laptop or desktop with the necessary softwares to be installed. It would be useful if you do the hands-on. Thank you very much. I thank the I thank all the organizers and RUSA team, our